Good afternoon, everyone.
Good afternoon.
Hi, and welcome back. Nice to see you on the virtual screen here.
I don't know if you can hear me. I can't hear you for some reason.
Yasmin, I can hear you. Let me type into the chat window. Are you hearing me okay, folks?
Yes. Just an FYI, Dr. Allen, Tiffany's going to be late in the class.
Okay, thank you.
Mhm.
Okay, so
I can hear you.
thank you for letting me know. So Yasmin, your audio problem is on your end because others can hear me. Okay, so let's see here. Okay. Just a moment, folks, while I help Yasmin out. Okay. All right. Okay, folks, welcome back. I hope you're doing okay. Lots of things to accomplish on a typical Thursday. I will, at some moment today, I will open the floor to you for questions about your lecture exam coming up a week from today. Okay, so we will do that. You will have opportunity to ask questions. But before we do that, let's be sure we finish the content off that we need to cover so that you have the information to do well on the exam. Okay. All right. So I'm going to share my screen with you and get back into conversation in chapter 13. Okay. And here we are. Okay. All right, gang. So, all right, we're going to continue our conversation. We had just started talking about HIV, the human immunodeficiency virus, and we need to talk about what it is in terms of its structure a little bit. And then we also need to talk to you about, well, how does the virus infect the cell? How does it replicate? Those sorts of topics. Okay, so I'm thinking maybe 30 minutes or so on this topic before we can pause and open the floor for questions. Okay. All right. So again, I'll reference you in your textbook. I hope you have it open in front of you that you can reference it. The best figure to understand HIV in your textbook is in fact figure 13.19, figure 13.19. And I would encourage you to have that open and available. Elena, I see your comment. Thank you. Okay, so let's try to get I have to do the same thing. I got to get my thinking cap back on here. Okay, so from last time, we told you that HIV is a retrovirus. And what that means is that HIV's genome is made of RNA. It is not made of DNA. And so part of the HIV life cycle involves taking the RNA and reverse transcribing it back into DNA. And as we get into this, it will become apparent why that is important. Okay, that is important because of something HIV as a virus actually does to our own host cell DNA. Okay, so let's continue at this time. What I'm going to do is give you a quick laundry list of some important molecules that are part of HIV structure. I'm not going to tell you everything, but I'm going to give you a list of molecules that are important for understanding the infection and replication process. So this will be a list of molecules important for HIV infection and replication. Okay. All right. So first off, like all good retroviruses, HIV inside its capsid contains 2 identical copies of plus strand RNA. Remember, plus strand RNA refers to sense strand RNA. And as such, when that RNA is dumped into the cytoplasm of a host cell, that RNA can be immediately used as a template for the host cell ribosomes. Okay, so HIV's capsid contains 2 copies of plus strand RNA. In addition to that, there's going to be other molecules we find inside the capsid, inside the core of the HIV virion particle. Additionally, you will also find multiple copies of a reverse transcriptase enzyme. Remember, the purpose of the reverse transcriptase enzyme is to take the RNA and go backwards in the central dogma flow chart from RNA to DNA. All right, so inside the capsid, we're going to find 2 copies of plus strand RNA. We're going to find multiple copies of reverse transcriptase enzyme as well. Let's see here. What else should you know for this topic? Okay, on the surface of the capsid, there is a special protein called GP120 that's on the surface of the HIV virion particle. GP120 stands for glycoprotein with a molecular weight of 120 Daltons. Okay. So on the surface of HIV, there is a protein, there is a glycoprotein called GP120. Okay. GP120 is part of the viral anatomy, if you will. It sits on the outer surface of the virion particle, and GP120 is the molecule that the virus uses to grab onto molecules on the surface of the host cell. Okay, so GP120 is used to bind host cell proteins. Okay, so let's talk about that a little bit. Okay, when GP120 attaches to a host cell, it typically attaches to 2 molecules on a host cell simultaneously. Okay, GP120 binds to 2 molecules simultaneously on a host cell. Okay, those 2 molecules are first CD4 receptors. Okay, CD4 receptors. CD4 stands for cluster of differentiation 4. It's an immune system molecule that's found on animals, on the surface of animal cells. Okay, so GP120 is a human cell protein. okay? And then there are other proteins, other receptors that HIV is potentially capable of binding. And so my next statement might seem a little bit weird, but stay with me here, okay? So the second molecule that GP120 will attach to will be one of two possible molecules, okay? If HIV infects macrophages, which you remember are a type of white blood cell, a defense cell that can perform phagocytosis, then the additional molecule is going to be a protein called CCR5. Okay, so CCR5 on macrophages, or the second possible molecule is something known as CXCR4. CXCR4, which is a different protein that's found on the surface of T cells. Okay, all right. Couple more molecules I need to tell you about here. Also inside the HIV capsid is an integrase enzyme. The purpose of the integrase enzyme is to insert a DNA copy of HIV into our human DNA. That's a really, really bad outcome. We don't want that to happen. But the enzyme that allows HIV to do that is called integrase. This enzyme functions to insert a DNA version of HIV into the host cell DNA. And finally, another molecule I need to tell you about is an additional enzyme also inside the HIV capsid. Is an enzyme called a protease. Okay. The purpose of the protease enzyme is to facilitate the maturation of immature, brand new HIV baby virus particles so that they can assemble correctly into mature virus particles, leave the host cell, go off and infect other cells, and continue the trauma of the body that HIV is known for. Okay, so the HIV capsid contains a protease enzyme, which is involved in the assembly of new HIV particles. Okay, so we have a protease enzyme here. We have an integrase enzyme here. Okay, so let me summarize what I've just stated, okay, because there's a laundry list of topics here. Inside the capsid of HIV, you will have 2 copies of plus strand RNA. You will also find in the capsid a reverse transcriptase enzyme. You will also find in the capsid an integrase enzyme, and you will find inside the capsid a protease enzyme. Are there some other things? Yes, there are, but this is enough of the story so that you can understand what we're talking about. So those things that I've just stated verbally are in the capsid. On the surface of HIV, we have a single molecule that is of importance for our discussion today, and that is GP120. That's a glycoprotein, 120 Dalton molecular weight. Its job is to act like the hand on your arm to grab things. GP120 enables the HIV virus to grab proteins on the surface of the human cells. Well, what proteins does GP120 grab? It will always grab CD4 receptors. It will always grab CD4 receptors on any kind of cell. And in addition to that, depending on the category of cell HIV is attempting to bind to, if it's a macrophage, GP120 will bind to CCR5. Alternatively, If HIV is trying to bind to a T cell, remember a T lymphocyte for our A&P graduates, then HIV will attempt to bind to CXCR4, which is a co-receptor, okay? All right, so do you have any questions at this moment about the molecules that have been put into your notes? Okay, so to make sense of all this business, now a cartoon's going to help us, right? So a cartoon's going to show us all these molecules, it's going to identify them, show you how HIV does its thing. I'm going to run a video with you, let you watch it. Okay, the video is linked in your class notes here in this document. So it was also in your previous notes from last class that I didn't get to show you. We'll watch the video. We will step through this process together in our notes. Hopefully that will get you up to speed in understanding HIV. Okay, folks, let me see if I can get this loaded up here. Come on, computer, do something useful. Okay. Okay, so here we go. Excuse the little bit of commercialism we have to sit through to get a video here. Okay, here we go.
AIDS is caused by the human immunodeficiency virus, HIV. This is an enveloped virus. The envelope is derived from the host cell membrane with viral glycoproteins such as GP120 and GP41 inserted into the membrane as the virus leaves the cell. The core is composed of P24 capsid protein, and the P17 matrix protein helps maintain viral structure. Inside the core are two identical copies of single-stranded RNA viral genome and three enzymes, reverse transcriptase, RT, protease, PR, and integrase, IN. To establish infection, HIV must first attach to its host cell. Attachment occurs by interaction between GP120 on the surface of the virus and the CD4 antigen receptor on the surface of the host cell. In addition to the CD4 receptor, there must also be a co-receptor on the host cell. The co-receptor differs for different host cell types. In T lymphocytes, the co-receptor is called CXCR4, whereas on macrophages, the co-receptor is called CCR5. After attachment, the viral envelope and host cell membrane fuse, resulting in entry of the virus into the cell. Once the RNA is released into the cytoplasm of the host cell, reverse transcriptase makes a DNA copy of the viral RNA genome. As the DNA is being formed, reverse transcriptase degrades the RNA strand. A complementary DNA strand is then added by the reverse transcriptase, and the ends of the resulting double-stranded DNA segment are joined non-covalently. Treatment with nucleoside analogs or reverse transcriptase inhibitors interferes with these steps. The resulting circular DNA is then moved to the nucleus and inserted into the host cell chromosome by the viral integrase, IN, enzyme. The integrated viral DNA is now referred to as proviral DNA. Following integration, the proviral DNA may remain dormant, or with host cell activation, RNA may be synthesized from the DNA, yielding messenger RNA and viral genome RNA. Viral messenger RNA is translated, yielding viral enzymes and structural proteins. Some of the functional proteins are formed by cleavage of a long polyprotein by the enzyme protease. Protease inhibitors interfere with this step. GP41 and GP120 are inserted into the host cell membrane, and the structural proteins surround the viral RNA to form the core. Finally, the virion is released by budding.
Okay, so that was a little bit of an abrupt ending, but it did complete basically the description of the HIV replication cycle that I need to cover with you. So you have access to the video. In your notes, the YouTube link is there. And while I just get organized again to actually take notes, are there any questions you'd like to ask about the video that you just saw? Okay, folks, well, let's see what we can do here. Okay, so what I'm going to do is put an additional page here in the document so that I can begin drawing on the next page. Okay, so what we're going to do, and I encourage you to draw with me at your home. If you have some colored pencils or pens, have a nice big clean sheet of paper. We are going to try to trace out some major key ideas of the video that was just shown to you, and also highlight some of the molecules that have been put in your notes at this time. Okay, so when a person is first infected with HIV, science has discovered that it is possible that HIV can go out and infect a variety of different types of cells in the human body. That definitely occurs. But from a statistical point of view, the most common type of human cell that seems to be first infected with HIV is in fact a macrophage. So we're going to approach this conversation from a new infection of HIV getting into the body and trying to infect macrophages. Okay, that's what we're going to work with here. All right, so please give me a little artistic license because I'm not very good at drawing. Okay, and we'll do it this way. Okay, so this is going to be HIV infection of a macrophage. Okay. All right. So you saw from the video that you have an HIV particle, right? And on the surface of HIV is this GP 120 protein, glycoprotein 120. Obviously, I am not drawing things to scale because an individual glycoprotein is going to be quite a bit smaller than the overall virus. But again, I'm not much of an artist, okay? So we have the glycoprotein on the surface of the virus particle. And since this is a macrophage, okay, that macrophage, I'm just going to draw a little bit of the border of the macrophage cell. That macrophage is going to have two different kinds of proteins sticking on its surface. It's going to have CD4, and it's going to have a related protein right next door to it, which for macrophages is CCR5. Okay, so we have a virus particle with GP120 sticking outside the viral envelope, approaching two proteins on the surface of the macrophage. Okay, so here is the macrophage plasma membrane. Okay, GP120 will touch base. It will touch amino acids that are part of CD4 and CCR5 at the same time and get a firm lock onto the surface of that host cell. As you saw in the video, once GP120 attaches to these host cell proteins, two at a time, the virus will be internalized. Okay, so the virus enters the host cell, and when it does so, the viral capsid literally disintegrates and kind of dumps its cargo into the host cell cytoplasm. Okay, so let's do it this way here. So viral capsid falls apart and releases cargo. It releases cargo. All right, so at this point, Let me ask you a question in the chat window. Okay, question in the chat window. Here we go. Tell me the name of any molecule that HIV will deposit into the host cell cytoplasm. Okay, go back in your notes, think about the animation we just showed you. Tell me the name of any molecule that HIV will deposit into the host cell cytoplasm. Okay, all right, thank you, Rachel. Thank you, Yasmin. Okay, good. All right, good. Just so I know you're alive out there, right? I can't see you, so I don't know, if you're following along or not. So absolutely, viral RNA is going to get deposited into the cytoplasm, and these enzymes we put in your notes, reverse transcriptase, integrase, protease enzymes in multiple copies are going to be dumped into the host cell cytoplasm. Okay, please let me know if I'm confusing you along the way. Okay, please ask questions. So continuing here with our little cartoon, what you're going to have is viral. You're going to have the HIV single strand RNA, right? HIV single strand RNA, that's what it is. Okay, and you're going to have the reverse transcriptase enzyme, which was also dumped inside the cytoplasm. Sit down on this, go across it, and make a copy of HIV single strand DNA. Okay, so this will be the reverse transcriptase enzyme. Okay, so reverse transcriptase makes a single strand copy of the HIV DNA. It takes the RNA and reverse transcribes it into DNA. When that reverse transcriptase reaches the end of the RNA, it will stay attached to the DNA that it just made, and it will do a U-turn, like you do a U-turn on the street. The enzyme will come back. Okay, the enzyme will be sitting here at this point. It will do a U-turn, and it will come back, and it will make the opposite complementary strand of DNA. So at this point, we now have a double-stranded copy of RNA in a DNA format. Okay. So reverse transcriptase makes a double-stranded DS, right? Makes a double-stranded copy of HIV. Okay. Any thoughts, questions, or problems at this moment? Okay. You saw in the video that then this double-stranded DNA molecule, which is HIV in a DNA version, will gently circularize. And then the video said that the HIV DNA will move into the nucleus. Well, folks, it doesn't just float into the nucleus. There is machinery in the cell that will bind to the HIV DNA and look at certain information that's encoded in the DNA. Within DNA, there are these sequences of nucleotides called nuclear localization sequences, nuclear localization signals. And basically, it's kind of like you have a tow truck come up to a car on the side of the road, and the driver gets out of the tow truck, he goes back to the car, he hooks up the car behind the tow truck, and he looks at a note that's been written on the windshield. It says, tow this car to the local gas station. And so the driver gets in the tow truck and pulls the car along to the closest gas station it can find. What I'm saying is there are machines inside the cell that will bind to foreign nucleic acid in the cytoplasm, and it will interpret information in it, and it'll say, oh, I guess you want me to deliver this to the nucleus. Obviously, your host cell doesn't want that, but the virus does want that, okay? So machinery of the cell is going to move the HIV into the nucleus, and I'm gonna draw the nucleus at this time. Okay, so let's use purple here. So down here, I'll put in a purple colored nucleus. Again, not drawn to scale in any way. Okay, so inside that nucleus, this is a macrophage, right? It's a human cell, it's a diploid cell. There's going to be 46 chromosomes. So there's all kinds of human DNA inside this host cell. And I'm not going to make an attempt to draw all of it. That would be insane. Right? But here's your human DNA. So the machinery of the host cell is going to interpret the nuclear localization signals on the HIV double-stranded DNA. And it's literally going to attach to that cargo, and it's going to pull it into the nucleus through a nuclear pore, which you might remember from your eukaryotic cell biology studying. Okay, so the virus is put into the nucleus. Okay, and I will encode the virus at this point as a double-stranded blue thing. Okay, oh, come on. Well, I'll stick with green since the computer wants me to use green. Oh dear. Okay. Be nice if technology would be helpful. All right. So we have a little snippet here of HIV DNA. Okay. Now, when the HIV DNA is brought into the nucleus, unbeknownst to us, and perhaps not as clear in the video as we would want it, there's a special protein attached to the double-stranded version of HIV, and that's the integrase enzyme. Remember, because we're going to have reverse transcriptase, integrase, protease enzymes dumped into the host cell cytoplasm when the HIV infects the host cell. So we're going to have, and I'm kind of running out of colors here. Let's see. We're going to have this integrase protein in here at the same time, which originally came into the cell via the original HIV virus that infected the cell. Long story short, integrase is an enzyme that has the ability to cut the nuclear DNA of the human cell, to cut it, to separate it into two fragments, to grab onto the HIV DNA, stick it into position, and glue it back together. Okay, so this enzyme has the ability to integrate the viral DNA into the host cell DNA. And again, I'm not much of an artist, but when this happens, the result is going to be something like this. Okay, you're going to have the human DNA, and it's going to be joined, it's going to be linked together with the viral DNA. Okay. So HIV has now become a physical component of our human DNA, of our human chromosomes in our human cells. Okay. HIV is now a physical part of our own DNA. Okay. I have a question for you that might seem a little silly, but humor me in the chat window. When you are watching a television show, how do you typically know something bad is about to happen? Okay, thank you, Chelsea. Chelsea must be the new Kit because she was sitting on her keyboard, wasn't she? Okay, so absolutely, right? You get these ominous, suspenseful sounds as music that alerts you that something in the storyline is going to go bad in just a second or just a moment, right? Well, my point here is this. In our cartoon here, when HIV has successfully integrated into your human DNA, the ominous, suspenseful, eerie music is absolutely raging. It is raging at this moment because now the virus has established a beachhead into our own genome. Okay? Now, let me ask you another question in the chat window. What did the animation call the integrated version of HIV? What was it called at that point? Once it was in our DNA, what was it called? Started with the letter P. Do you remember? I know you only saw it once. Okay. Not a protease, Selena, but thanks for trying. Okay, so let me help you out here a little bit. You may recall that it was called a provirus, okay? Let me type that into the chat window. A provirus. So when HIV has successfully inserted itself into our human DNA, it is now called a provirus, okay? And so let me just use that word here in the drawing. Here's another question coming in the chat window. A provirus sounds similar to an idea we talked about when discussing bacteriophage lambda last time. What was that term? It was not provirus. What was it? Do you remember, folks? I know this is ancient history, right? It was 2 days ago. Okay. All right. Thanks so much, folks, for taking a look at that, right? You're absolutely right. The prophage is what I'm talking about, okay? So here's the deal. When HIV inserts into the human DNA, it becomes a provirus. When bacteriophage lambda inserts into the bacterial DNA, it becomes a prophage. Folks, there is a critical difference here. There is a critical difference here, okay? A provirus, once it inserts into the host DNA, it stays there forever and it never leaves. It never leaves, it never pops out, it never excises. Forgive the analogy, but it's probably like your mother or father-in-law coming to visit and never going home. Okay? That's what this is like. Okay? So provirus never leaves the host cell DNA. What I'm basically telling you is that when this macrophage reaches the end of its useful lifespan and the macrophage dies, it's going to die with a copy of DNA, with a copy of HIV in the DNA. because that proviral DNA stays there forever. Okay. You might recall, and I'm not going to put this in your notes here because I don't want you to be confused, but you might recall when we talked about lambda last time, we said that the prophage DNA, when it incorporates into the bacterial DNA, it can at times, depending on environmental triggers, actually pop back out. So a prophage can enter the DNA and exit the DNA. A provirus can only enter the DNA and it never leaves. It stays there forever. And what I'm saying here is that this should be the ominous music in our television show, because this is really, really bad news for us as human beings, because this virus is going to be in us for everywhere. Okay. So Yasmin, that's why it's so hard to kill the virus. Basically, yes, Yasmin, it is. Okay. All right. So at this point, what's going to happen next is that you're going to get gene expression of the viral DNA that's just incorporated, right? The video showed you that you were going to get transcription of the genomic transcription. Okay, I'm running out of colors here, but you're going to get transcription of this thing into genomic HIV RNA. Okay, that's the HIV RNA that will be put into new capsids of new HIV virus particles that will eventually leave the cell to infect other cells. And at the same time, you're also going to get transcription of the other side of the DNA helix of the provirus, so that you're going to get HIV messenger RNA made. And that messenger RNA then can be, you guessed it, it can be exported from the cell, where the HIV messenger RNA will be translated, right, by host cell ribosomes to manufacture new proteins, new enzymes that new HIV particles are going to need when they assemble together to form new HIV particles, right? So this HIV messenger RNA is going to be translated into brand new reverse transcriptase enzymes. Integrase enzymes and protease enzymes, among other things. Okay, so these are the new building blocks for new viruses to be assembled, for new virus particles to be created. Okay, and I'm running out of room here, but okay. Let me ask a question in the chat window. When messenger RNA is translated, what is manufactured? When messenger RNA is translated, what gets made, folks? Okay, thank you, Yasmin. A protein, absolutely, a protein, right? You know this from chapter 8. You know this from chapter 8. And what I'm telling you here, though, is in chapter 8, you would have gotten the impression that typically you have one piece of messenger RNA, a ribosome translates it, and you get one protein. One messenger RNA, one protein. That's the basic idea. But Mother Nature sometimes doesn't obey that rule. Sometimes Mother Nature will take a long piece of RNA and actually manufacture a huge protein that has multiple segments connected together. So in the video, you might have caught the phrase polyprotein, polyprotein. So what happens here is that this HIV messenger RNA that comes out of the nucleus, it codes for multiple proteins. So the protein that initially gets made has multiple segments to it, and I'll indicate that with vertical bars. Okay, so here's the polyprotein. So the first segment of the polyprotein might be the reverse transcriptase enzyme. The middle segment of the polyprotein might be the integrase enzyme. And the third segment of the polyprotein might be the protease enzyme. Okay, so for these proteins, which are glued together to do anything useful and have physiological activity, they must be cut apart. They must be cut apart. And so they will be cut apart from the protease enzymes that came in with the original virus infection. Okay, so down here, we're going to have some protease enzymes. I'm sorry this is getting so crazy and so busy, but this is really how the cells do their thing, right? So this protease enzyme down here that came in from the original virus particle, it's going to float over here, and it's literally going to cut at these vertical segments to separate the newly made polyprotein proteins, so that the new reverse transcriptase enzyme is separated from the new integrase, is separated from the new protease. And these things will all be used together with the genomic RNA, which I've still drawn in the nucleus, but which honestly eventually gets exported out of the nucleus. All of these things, plus some additional molecules we haven't talked to you about, are going to come together inside the cell and be used to build brand new virus particles. Okay. I am out of space here, as you can see, but all of this here will be used to make new HIV particles. Okay. Before I go any further here, because I don't want to overwhelm you, but at the same time we have more to do. Before I go any further, are there any questions you'd like to ask about this? Okay. All right, please let me know if you do. Okay, so let me just summarize everything I've done here quickly, and we will move forward. When HIV infects a macrophage, the glycoprotein 120 on the surface of the virus binds to two proteins on the surface of the macrophage called CD4 and CCR5. That binding event triggers endocytosis so that the virus is brought into the cell. When the virus is brought into the cell, the virus structure kind of falls apart. And the reason it does that is because there's localized changes in pH, but who cares right now? So the virus particle is brought into the cytoplasm, the capsid falls apart, dumps its cargo into the cytoplasm. The viral reverse transcriptase enzyme is going to sit down in the HIV genomic RNA and eventually build double-stranded HIV in a DNA form. That double-stranded DNA then enters the nucleus of the host cell along with the integrase enzyme that came in with the original virus particle. The integrase enzyme cuts the human DNA and sticks a copy of the HIV DNA right in between our segments of DNA. HIV has now become a provirus, and cue ominous music, it will stay there forever and never leave. That HIV proviral DNA will be transcribed. Both strands of the HIV will be transcribed into genomic HIV RNA and HIV messenger RNA. Those things will be exported out of the nucleus. The HIV messenger RNA will be translated into a giant polyprotein by the human cell ribosomes. And then protease enzymes that came in with the original virus will cut the polyprotein apart so that I have the individual subunits I need to assemble new HIV virus particles, which will then exit the cell. Any questions? Okay. All right, so I'm just going to take up shop now and try to do some Q&A with you here. Come on, software, work with me. Oh, sorry. Oh, come on. All right. Questions and comments here, okay? All right. Okay, so let's see what we can do. Okay. First thing I want to say to you is that when new virus particles are manufactured in the host cell cytoplasm, well, let me ask you in the chat window. Oh dear Lord. Okay, here we go. Here we go. When new HIV particles are assembled inside the host cell cytoplasm, do the new HIV particles have an envelope? Okay, very good, Taylor, Yasmin, Geneva. They don't, right? They don't have an envelope yet because they're not going to acquire the envelope until they push out of the cell through an exocytosis-like process. Okay, so that's the first thing I want to comment on in here. The new HIV particles inside the host cell acquire their envelope only when they push through the plasma membrane of the host cell. Okay, so when they're just newly manufactured, it's just a capsid with all its cargo inside, but until it actually pushes through the plasma membrane of the host cell, it doesn't yet have an envelope. Okay, next. Okay, we need to talk a little bit about, well, okay, if we understand the basic idea of how HIV infects the cell and replicates inside the cell, we need to start connecting this to, well, how does all this lead to the development of AIDS? Okay, so I'll put this down as a question. Okay, how does this replication process explain how and or why AIDS develops? Well, here's the basic idea, folks. Okay. The reverse transcriptase enzyme, okay, up here, which I've written in green, the reverse transcriptase enzyme is very, how should I say it? This reverse transcriptase enzyme of the virus is extremely error prone. It tends to make mistakes, okay? So that's the first thing. The HIV reverse transcriptase enzyme is error prone. It has a, in comparison with similar polymerase enzymes, HIV polymerase makes an unusual amount of errors, okay? As HIV reverse transcriptase takes the viral RNA and decodes it back to DNA, there's a good chance a mistake is going to be made. And as HIV copies itself inside a host cell and builds new virus particles that then go out into subsequent cells and they repeat this process, we're performing reverse transcription many, many times as we infect multiple rounds of cells in our tissues of our bodies. So what I'm saying is that we're doing reverse transcription a lot as we infect throughout the fabric of our body. Eventually, reverse transcriptase is going to make a mutational change in the viral sequence that causes brand new virus particles that are made to have a slightly different modified GP120 glycoprotein. Eventually, a mutation created by an error-prone reverse transcriptase enzyme causes new HIV particles to be manufactured with a slightly mutated GP120 glycoprotein. Okay, so maybe you can see now where I'm heading. Okay, so I'm going to ask a question in the chat window. Question in the chat window. Here we go. What does the mutation in the GP120 glycoprotein enable the new HIV particles to do? Okay, Rachel, you say attached to the host, but the original HIV particles could attach to the host too, right? So how's that different? Anybody have a thought on this? Okay. Yasmin? All right, everybody take a look at Yasmin's comment in the chat window. She says, attached to the CCRS and CD4. So Yasmin, you're starting to get at it. You haven't quite identified the molecules correctly, but you have the right idea, okay? So remember the following. We'll put this into your notes. Remember the original HIV GP120 bound to CD4 and CCR5 on the macrophage. Okay, you remember that. When you have a mutated GP120, will bind to CD4 and CXCR4 instead. Okay, so let me ask in the chat window. What type of cell in your notes has a CXCR4 co-receptor? What type of cell in your notes has a CXCR4 co-receptor? Okay, we put it in today's notes. Okay, thanks, Aubriana and Ireland, right? T cells. Okay, T cells, absolutely. Okay. So I'll finish the notes here. The mutated GP120 will bind to CD4 and CXCR4. Instead, CXCR4 is only found on T cells. So here's what I'm telling you. Early in the infection of HIV, early in the replication process, HIV will predominantly grow and replicate inside macrophages. And that can go on for months to years, depending on the personal health status and genetic makeup and strength of that person's immune system. That process of replicating inside macrophages can go on for months to years. And then at some point, it's like a switch has been flipped, right? Click, light on, light off, click. It's like a switch has been flipped in the sense that all of a sudden, HIV starts to infect T cells rather than macrophages. Well, why did that happen? It happened because in the process of replicating, the reverse transcriptase created a mutation which modified GP120 in such a way that now GP120 prefers to bind to T cells rather than macrophages. Okay. All right, so let's see here. Letter C. Okay. When HIV infects and leaves macrophages, macrophages are not killed. When HIV infects and leaves macrophages, macrophages are not killed. So HIV is viable inside the macrophage as long as the macrophage is alive. That macrophage becomes a production platform to manufacture new virus particles, and there's not a single bit of thing we can do about it. I mean, yeah, we do have some medications that kind of suppress viral replication, and that's helpful and useful, but we don't have any medications that can actually remove the virus from our DNA and literally cure us. Okay. You may have heard the occasional story about, somebody got a stem cell transplant, they seem to be cured from HIV. Yeah, that has happened a couple times, but we don't understand why it worked and we can't replicate it. Okay. So long story short, when HIV infects and leaves T cells, right, T lymphocytes, the T cells are killed. That is a critical difference. Macrophages are not killed by replicating the virus and spitting them out. T cells are killed. Okay. So I don't know how much immunology you have in your background, but for those that remember some immunology, there's basically two branches to the immune system. There's the antibody-mediated immunity that deals with antibodies, and there's a cell-mediated immunity that deals with particularly T cells, T lymphocytes. So when HIV starts infecting the T cells and killing the T cells, you're basically cutting your defense force in half. And so when that happens, all of a sudden your immune system gets weaker, okay? All right, so death of the T cells causes a decline in the health of your immune system, which means that you have now acquired immunodeficiency, right? AIDS, acquired immunodeficiency syndrome. So that means when the T cells are dying, when they've died, your immune system is no longer capable of protecting you from everyday normal threats, such as cold viruses, or maybe a strep throat infection, things that we kind of slough off and say, oh, I can handle that. Yeah, maybe I need an antibiotic for strep throat, who knows? But I can survive it, I can live it. But a person that is actively in the process of developing AIDS, that has AIDS, their T cell count is so low that their body effectively cannot mount any new responses to other pathogens, and those pathogens overwhelm that person's body, and particularly without medical care, that person will quickly succumb and die and pass, unfortunately, from AIDS. Okay. All right, folks. That was longer than I wanted it to be, but I got it done. Are there any things you'd like to ask about this? I mean, you appreciate that in terms of the coronavirus pandemic that we're all living through right now, modern biotechnology tools, gene engineering, all this kind of stuff enabled primarily Pfizer and Moderna to design and manufacture an effective vaccine in less than a year. And that has had a dramatic impact on the number of people that have passed away due to coronavirus. So in less than a year, a vaccine was pushed out to the community, and ignoring any arguments of politics and things aside, a vaccine was available and it has been effective, and it is effective. HIV, the earliest reports of HIV infections go back to publications in the late 50s or 60s. We as a civilization have been attempting to design and develop an effective HIV vaccine for upwards of 4 decades, and we still do not have one. We do not have an HIV vaccine. There are just aspects of the HIV replication process and how it interacts with our own bodies and our immune systems that we do not understand sufficiently well to design a vaccine, any vaccines that are effective. There have been many attempts, many failures. There have been clinical trials. There have been clinical trials with HIV vaccines where things have been attempted, where the actual vaccine was developed when it was given to test participants in the clinical trial. The presence of the vaccine actually made it easier for the virus to infect the host person. Okay, so some of the vaccines had the exact opposite effect of what we thought they were going to do. So it's a problem. So Yasmin, you ask, is it because it happened so fast? Honestly, Yasmin, I don't think it's because it happened so fast. I think it's because we don't understand enough about the underlying molecular biology of the virus's interaction with our human body, that so far, no vaccines have been successful. So, you know, a year for a coronavirus vaccine, that's amazing. 4 decades for an HIV vaccine, we don't have it. People have been trying this, you know, even before I started graduate school in the Stone Ages, people were trying to deal with HIV and nobody's been successful yet. Okay, folks, do you have any questions about HIV? All right. So here's where we'll spend our time. I want to open the floor to you in terms of your study guide. When we get to lab today, I think I am going to do a little bit of lecturing on chapter 20 because I need to get that started. 20-30 minutes on chapter 20. We'll get lab stuff done like we normally do, and we'll be on our way, okay? So let me open the floor to you. I'm now done lecturing with any content that will be on lecture exam 2. So you know lecture exam 2 is a week from today on the 21st. You also know that this coming Tuesday is Tuesday of your midterm break, so we do not have class. Okay, we won't be together. I know everybody sheds a tear on that. We will not be together next Tuesday, so I can't answer your questions next Tuesday. So now's really the time to ask questions. Is there anything that you would like to ask about exam 2? Let me bring up the study guide that was sent out to you. Okay, so this was sent out on Tuesday's email. I hope you had a chance to download it and at least read through it once. Is there anything you'd like to ask in terms of the study guide here so that there's no shock? There's no shock next week when I ask you to do things.
Professor, can you elaborate on 4B?
Okay, all right, 4B. Here it is. It says, describe a specific example of an environmental circumstance that will cause a specific gene to be expressed and a specific example that will cause a specific gene to be repressed. The example that we talked about in class, Selena, was the lactose operon. So in the lactose operon, the specific example of an environmental circumstance that would turn genes of the lactose operon were the absence of glucose, and the presence of lactose. There's your environmental circumstance. The presence of glucose, the absence of lactose, excuse me, I'm going too fast. The absence of glucose and the presence of lactose would turn LAC-Z gene on. Okay, there it is down here in the study guide. The presence of glucose and the absence of lactose would turn the LAC-Z gene off. Do you remember that, Elena?
I do.
Okay, so that's my example to you. That's what we talked about in class. And what I'm saying is, if you understand, the basic idea that I want students to get is certain environmental triggers can turn genes on and certain environmental triggers can turn genes off. I think you already understand that philosophically. But now what I want you to do is find any kind of circumstance in all of biology, not necessarily microbiology, that you're interested in, and give me an example. Maybe you're interested in genes that get turned on or off during a disease process, like multiple sclerosis. Maybe you're interested in getting an example of genes that are turned on or off during the menstrual cycle, or during the production of sperm, or during the flowering of a tree. Whatever it is, I don't care. What I want you to do is find me a specific example where you can cleanly identify an environmental circumstance that turns the gene on, and then usually the environmental circumstance that turns the gene back off is to remove that environmental trigger. So I want you to find an example that you're interested in and document it for the exam. All right, Elena, does that make sense?
Yes, thank
Okay.
you.
All right. Are there other questions you'd like to ask about?
Yes, for 6, are we gonna write like the whole DNA, then the mRNA, and then the whole protein, correct?
That's the basic idea, and I will say to you that the exam is set up. The exam is set up to guide you to through the proper path of what you need to achieve. Okay.
Okay, so you would give us the DNA, or we would figure the DNA out.
Well, here, take a look at what it says here. Set 6
Oh,
says,
okay.
given a starting sequence of DNA. So of course, I will give you a starting sequence of DNA.
Okay.
Okay.
Thank you.
Let me highlight some information about the unit conversions, right? I know that there's been some anxiety about math. And while I can't remove the anxiety from you, please know that preparation and practice of this skill helps reduce your anxiety. Okay. On your exam, what you're going to have to do is do the unit conversions, and I want you to write out your work on paper. And after the exam is over, I want you to take a picture or make a PDF with the Adobe Scan app, or maybe some of you are using Cam Scanner, I don't know. Okay, so either through a photo or a PDF, I want you to send me documentation of your handwritten work. Here's my email address right here in the study guide. I need to receive that email within 15 minutes after you complete your exam. So you're going to be given 1 hour and 25 minutes to take the exam. After 1 hour and 25 minutes, the stopwatch will begin for an additional 15 minutes that you have to scan your work and email it to me. When I go into class marker and I'm grading your exam, I'm going to be taking a look at your unit conversion answers, but then I'm going to be going back to your email and looking at your workflow and seeing if your workflow actually justifies the answer that you gave me. Okay, so I'm going to be making sure the answer is justified by the work.
And you want that emailed to you, not put into like our normal lab Dropbox.
Yes, Rachel. In this case, I want it to be emailed to me. That way, if I need to make specific comments back to individual students, I'll already have an email to go right back to. It just makes it a little easier.
Okay, thank
Okay.
So when you say essay, Dr. Allen, so are we having something similar to what you had provided us on the worksheets?
Yes.
Okay,
I will present a silly little scenario to you that will test your ability to do unit conversions.
Okay.
All right. Okay. Is there anything else that you would like to ask about? So we're at the end of our lecture period here. So we'll take our little break, come back to lab. Let's start lab. Let's start lab at 2 o'clock. That's about 14 minutes away. And I'll start by asking you again if you have any questions for the exam. Otherwise, I'll start some new content in chapter 20. Okay. So Zoom channel will stay open. Take a little break. Think about if there's any questions you have, and we'll get started at 2 o'clock. Okay, hi folks, welcome back. Hope you had a chance to get a little break, maybe get a snack or something. And let's see if we can reconvene here into a common space and continue our work for this afternoon. Okay, so just one more time for the benefit of anyone. Are there any topics you'd like to talk about for lecture exam 2 coming up next Thursday? Okay. All right, folks. Well, please let me know how I might help you. Next Monday and Tuesday, of course, is your midterm break. So I really do hope that you take a little bit of time for yourself. But obviously, I also kind of hope that there's some serious study going on. On Monday and Tuesday, I will not hold office hours. I will not be available to you on Monday or Tuesday. However, Wednesday, I'll be back with office hours Wednesday before the exam. And next Thursday morning, I have office hours as well before the exam. So there will still be 2 office hour sessions that you could come in and talk with me if you're having problems. Okay, so let me know how I might support you.
I actually have a question. For the first one, for the essay, it says like to list the characteristics of any virus. Do we write what virus we're talking about? Or just,
Yeah, okay, Yasmin, it's not necessary to identify a specific virus. Okay, you can just make 4 general characteristics of viruses.
okay.
Anything else? Okay, well then, folks, good luck to you. Lecture exam 2 is on chapter 8, genetics, chapter 13, viruses, and the unit conversion skill that you've been wrestling with for several weeks now. Okay, you can do it. I have faith in you. I have confidence in you. Okay, I'm going to start talking about chapter 20 at this time, just so we don't get really behind. So next Thursday will be your exam. And as I mentioned in the study guide, right, I mentioned in the study guide that because I'm giving you additional time on the lecture exam next Thursday, that means we'll start our lab next Thursday a little bit later than normal, right? Lab next Thursday will start at 2:15 to give everybody a chance to finish their exam, shoot me off an email with their unit conversion work, and maybe get a little bathroom break in between, okay? So just keep that in the back of your mind that lab next week will start at 2:15. Okay, so what I'm going to do is start talking about chapter 20 content. Chapter 20 is not on your exam this coming Thursday. So, we'll take some notes. You don't have to study them though for next Thursday. Just be aware that we're taking notes in chapter 20. Okay. The title
Professor,
of,
I'm sorry, Dr. Allen. Would it be feasible if you would start chapter 20 on a different paper?
Yeah, I can do that.
That way I could separate it.
No problem. We can do that.
Thank you.
Chapter 20, part 1. All right. Okay, folks, let's get started here. Okay, so the title of chapter 20 is antimicrobial drugs. And of course, we will be talking about antibiotics in this chapter, but you'll quickly come to see that antibiotics is not a catch-all phrase for everything. And it's often been bastardized in our common society that people don't even know what an antibiotic truly is anymore, at least from a technical perspective. We're going to talk about these things, okay? So let's define chemotherapy, okay? So let me ask you in the chat window, what topic enters your mind when you read the term chemotherapy? Right? I mean, if we're being honest, that the big C word, right? The big scary C word, okay? So chemotherapy is something that we think about with cancer, okay? I mean, that's what the common perception of it is. But from a science perspective, chemotherapy simply means treating a disease with chemicals. That's what it means, treating a disease with chemicals, okay? So chemotherapy refers to the treatment of any disease with chemicals. So that means treating cancer with some kind of cell cycle inhibiting drugs is chemotherapy. It also means when you take a Z-Pak to treat your sinus infection, that is also chemotherapy, okay? So chemotherapy is the treatment of any disease with chemicals. So this term is equally valid for treating cancer and strep throat, right? There really is no difference in terms of our understanding of what chemotherapy means. We simply in our society think, oh, cancer and it can't be anything else. That is patently false. So, okay. Let me ask you a question in the chat window. Oh, hold on, I gotta change who I'm sending this to. Hold on, here we go. When you purchase an over-the-counter cream at the local drugstore to treat your case of athlete's foot, is this an example of chemotherapy? Okay. Wow, that's an interesting response there. And take a look at the responses, folks. We have quite a bit of disagreement here. Okay. Well, folks, athlete's foot is an infection of the dermis of your skin caused by fungi. It's a fungal infection. Okay. So athlete's foot is a disease. It is a condition caused by a fungal infection of your skin. So when you apply medicated creams to try to kill the fungus that's causing the athlete's foot, it is absolutely an example of chemotherapy. It is chemotherapy. That's the point I'm trying to get across to you, okay? All right, let me ask you this. Have you ever had a real case of athlete's foot? Okay. All right. So most of you were no on that. Some of you were yes. For those no people, hey, next week when you're off a midterm break, I want you to go get a case of athlete's foot and see how it feels. Okay. All right. I'm teasing you. Okay. I don't really want you to get sick, but these things do happen, typically associated with sporting events and things like this. Absolutely. Okay. Okay, folks. So we now know what chemotherapy is. So this is a term that's valid for treating cancer and strep throat and even athlete's foot. Okay. Now, you know quite a bit already about some of the history of the development of antibiotics. Last week we had a video that showed you Alexander Fleming, and it talked a little bit about the discovery of the very first famous antibiotic, penicillin. Okay, let me just give you a few notes on that historical event. Okay, certainly this is not, I'm not a historian or anything like that, but it is useful to hear these things and be aware of the history from which the field has developed and itself evolved. So let's talk about discovery of penicillin, or what we think of today as penicillin, the very first antibiotic known to man. Okay, so penicillin was discovered, penicillin, or I should say, the effect of penicillin, not literally the molecule penicillin, but what penicillin was capable of doing. The effect of penicillin was first observed by Alexander Fleming in 1928. The effect of penicillin to kill bacteria was first observed / identified by Alexander Fleming in 1928. And, I hope you catch how I changed the wording of what I was saying to you, okay? In 1928, Alexander Fleming had no idea what the actual molecule, which is penicillin, was. He didn't know it back then in 1928. He simply had observed a curious result on a petri dish growing in his lab and thought to himself, right? That's kind of curious. What the heck is this all about? Okay, so the effect of penicillin to kill bacteria was observed by Fleming in 1928. Okay. The very challenging analytical work, scientific work to figure out exactly what it was in the petri dish of 1928, which was responsible for killing bacteria that Fleming had observed. The actual scientific work of figuring out what the structure of the molecule was took 10 years of scientific research. Okay, so it wasn't until about 10 years later in 1938 that we knew what penicillin was, and we started to have the capacity to actually manufacture it in large quantities. You noticed in the video, if you remember from last week, that penicillin was so important in the early days of its discovery that people that were treated with penicillin, they had their urine collected, and the urine was filtered for any unmetabolized penicillin that could be recovered, right? It was that important, it was that valuable that humanity went to great extents to collect urine from patients being treated with penicillin, so any unmetabolized penicillin could be recycled and reused. Okay, so it took another 10 years of research to identify the molecule now called penicillin and begin mass producing it. Okay, so I have a question for you in the chat window. What important worldwide event was getting started in 1938? Okay, now none of us were alive, right? But think about what you've learned in your history classes, in your history lessons, right? Okay, so 1938 is, we're talking about that time in civilization's history where we were on the brink of World War, right? The Second World War against Adolf Hitler and the Nazis, World War II. And so what I'm pointing out is there's a really historical significance to the development and manufacturing capability of penicillin, right? Penicillin was a highly regarded substance during World War II because it saved the lives of a lot of wounded soldiers that survived their initial trauma, but then were combating infection in medical hospitals and MASH units around the world. Okay, so this was a really big deal in 1938, 1939, and all the years of World War II. And that's sort of that mystique of getting wounded in combat and then going to a hospital and surviving an infection because you were given penicillin kind of created the fame of penicillin as the wonder drug. I mean, right, everybody, after World War II, they were saying, oh, we're never going to have to worry about infectious disease again. Now, obviously, that was profoundly naive. We will always have to worry about infection because Mother Nature constantly evolves around the medications and drugs that we work so hard to develop. But in that period of time, everybody thought, oh man, penicillin, I'm good to go. I'm going to be okay, right? All right. So penicillin, just to give you a little preview on this, if you have your textbook available and you can shift through it, take a look at figure 20.6. Take a look at figure 20.6. Today, penicillin as a molecule comes in many different varieties. Okay, but if you look at figure 20.6, penicillin G there, panel A, penicillin G as in George. Penicillin G was the version of penicillin that Alexander Fleming stumbled across during his laboratory experiments. Okay, so today there are many versions of penicillin. Okay, the version that Fleming stumbled across is penicillin G. And notice how your textbook labels this drug. It labels it a natural penicillin. And the point of it here is that penicillin is a molecule. Now, if we ask ourselves, well, what is penicillin? And how is it made? The honest answer is this. Okay, so here's point C, and I'm going to make it a question. What is penicillin and how is it made? Penicillin is a molecule that has the structure you see in figure 20.6, and it is made by in this case, a species of mold. And you remember from your studies from chapter 1 that mold is a type of fungus, right? So we have a mold, which is a type of fungus, which manufactures this molecule naturally. And this substance that the mold manufactures is pushed outside the cell into the surrounding environment to destroy bacterial threats to the mold. Okay, that's so in one way of thinking about this, this is a type of germ warfare. I do not mean the kind of germ warfare where humanity tries to make each other people sick with other germs. I don't mean that kind of germ warfare. I'm talking about Mother Nature's germ warfare, where one species of cell manufactures a poisonous substance which kills other cells that the first cell doesn't like. That is really what penicillin is, and that is how it is made naturally in the cells that Mother Nature has evolved and created. So penicillin is the molecule of figure 20.6. It is made by a mold of the name penicillin. Hold on a second, I gotta check my notes on this one because it's a weird name. Penicillium chrysogenum. Okay, Penicillium chrysogenum is a mold, and that is the mold that Fleming discovered on his petri dish, and later figured out that, oh, there's a molecule here that we're going to name penicillin based off this mold called Penicillium that is responsible for killing some types of bacteria. Okay. All right. So that's your quick history lesson on the discovery of penicillin. Do you have any questions? All right. So let's talk about what an antibiotic is. Okay, antibiotic. Okay. An antibiotic, literally, think about it. The prefix anti means against, against. And the ending of the term biotic refers to life. So against life. That's literally what this phrase means, against life. And in a bit more formal way, an antibiotic is a chemical manufactured by one microorganism to kill or inhibit the growth of a second different kind of microorganism. In the case of penicillin, okay, the mold, which remember again is a fungus, Penicillium chrysogenum, produced the molecule penicillin to kill surrounding bacteria. I've never read anything in the history books that tells me, what specific species of bacteria Fleming had on his petri dish plates. I suspect that might be information that's been lost to the history, to the years of history. But nonetheless, penicillin is a molecule made from the metabolism of a mold to kill bacteria. Now, why would mold do this? Well, the mold was doing this in the case of Alexander Fleming's experiments because the bacteria was literally eating the food source that the mold wanted. Okay, so let me ask you a question in the chat window. If your next door neighbor kept eating food out of your refrigerator, would you get upset? Okay. Of course you would, right? If your next door neighbor is brazen enough to enter your home and eat your food without even asking permission, without even contributing any money to purchase the food, it's not going to get long for you to get pissed off, is it? Okay. And so that's the scenario with this mold, right? The mold wanted the food in the surrounding environment of the petri dish. Bacteria was eating it. Mold didn't like that. Mold wanted the food in the petri dish. So the mold created a poison that we happen to call penicillin today to kill the bacteria so that the mold would have access to its food supply. That's what I mean by Mother Nature's germ warfare. Okay. All right. So antibiotic means against life. It's when a chemical is made from one type of microbe to kill or interfere with another type of microbe. In today's society, the term antibiotic has been hijacked to imply antibacterial. Okay, so if you go to the doctor because you have a strep throat infection, which is caused by a bacterium, Streptococcus pyogenes, they're going to give you an antibiotic. So, treating bacterial infections has become synonymous with the term antibiotic, even though technically it doesn't have to be treatment of a bacterial infection, right? We could have an antibiotic being used to treat a protozoal infection. We could have an antibiotic being used to treat a fungal infection. Okay. And so we have to be very, very careful today when we talk about antibiotic, what we're really referring to. Common everyday language suggests that it's antibacterial in nature, but it doesn't really have to be. Okay. That was not its original intention when the term was defined. Okay. So I'm going to try to be, in this chapter in particular, I'm going to try to be very over the top, clear cut, whether I'm talking about an antibacterial drug, an antifungal drug, an antiprotozoal drug, an antiviral drug. I'm going to say it that way. I'm not just going to say antibiotic. I'm going to say antibacterial, antifungal, antiparasitic. That way we know exactly what this drug is intended to treat, because antibiotic really refers to killing any kind of cell with something made from any other different kind of cell. All right. Thoughts or questions? Okay, so I don't want to spend too much more time here, but I would like to get in a very quick, maybe 5-10 minute discussion with you about the cost of drug development in the United States of America today. This is an appropriate chapter to do it because the topic is antimicrobial drugs, okay? Okay, today's cost of drug development in the USA. And we're going to share some numbers with you that may or may not surprise you. But at any rate, hopefully some of the things we're going to say kind of upset you, because it is upsetting. Okay, so is anybody, I'm going to just put a couple quick questions into the chat window here. Okay. What is the average time to bring a new drug to market in the United States of America? How long does it take a drug company to figure out a new drug and understand how it needs to be manufactured, to go through clinical trials and actually get it on the market? How long does it take? Okay, and obviously I'm talking in generalities here. Okay, there's some drugs that maybe got out there very quickly in terms of maybe 3 years it was out or something, and then there's drugs that have taken a super ridiculous long time. But the time frame I'm trying to suggest to you is that the average drug development time to get it out on the market for a new drug is about 10 years. Okay, it takes roughly, on average, 10 years to bring a new drug to market. Okay. Next question in the chat window. How much money is spent by a drug company to research and test a new drug before it can begin selling that drug? Yeah. Okay. The ballpark frame we're in here is well in excess of $1 billion. Well in excess of $1 billion. Okay. Research and clinical testing on new drugs requires a typical investment by a drug company of at least $1 billion US. Okay, so it takes big money and at least a decade of effort to get a new drug to market. And that's assuming that things go well, because sometimes they don't go well, right? All right, so let me ask you this, okay? Here we go. If you take a drug to treat any condition and it makes you sick or even kills you, how will your family likely respond? Obviously, if you pass away, your family is going to grieve, right? They're going to grieve because that's a very, very upsetting thing. And I truly hope that never, ever happens to you, okay? But medical things happen all the time, don't they? So the point I think that I'm trying to get at my question, and which many of you have indicated in your answers, is that once you get sufficiently past the grieving process, you're probably going to at least consider a lawsuit. You're probably going to sue, okay? So let's think about that for a moment, okay? If a person is harmed by a drug legally sold on the market, likely the drug company will face litigation. Okay. In other words, they're going to get sued. Okay. Let's think about this. Next question here. Okay. Coming in the chat window. Here it is. If a drug company has to defend itself against your lawsuit, who will the drug company pass the cost of defending itself off to? Okay. You see Rachel's comment. You. The point I'm trying to make is that if we sue drug companies, they have to legally defend themselves, which costs lots of money in legal fees. And so for that company to stay viable, to not go out of business, to not go bankrupt, they're going to have to increase the price of other drugs that they offer, which means they're going to pass the cost of defending themselves against litigation off to every other future customer of that company. Okay, so what I'm trying to suggest here is I am in no way, absolutely in no way, defending the high cost of drugs, because it is ridiculous and insane. But here's what I'm asking, and I want you to consider this carefully in the chat window. Here we go. Do all of us have to accept some responsibility for the high cost of drugs? Okay. Okay. I'm trying to get you to see is that the issue of high drug costs being ridiculously high is not a simple topic. It is not black and white. It's not black and white. You know, what would you expect a company to do? If you're suing it, the company has to defend itself. To defend itself, it needs money. how's it going to get money? It's got to sell other products that it has to increase the cost of that product to future consumers. So what I'm saying is that it's sort of a self-reinforcing cycle where we are somewhat forcing the hands of the drug companies to increase prices. Now, okay, so let me just put this down, and I'll just put it as a rhetorical question. Does all of society have some measure of responsibility for high drug costs? And I'm not going to answer that. I want you to answer it for yourself and think about that. But here's another question. Okay, here's another question, and I'll put this in the chat window. I'm actually going to put this one in your notes here. And I'm going to type and talk at the same time. Is it reasonable? Is it reasonable to expect any drug sold on the market to be 100% safe for every human being on the planet? Okay. What do you think, folks? Is it reasonable to expect any drug sold on the market to be 100% safe for every human being on the planet? I hope you can see that that is not a reasonable position, okay? And it's not a reasonable position because, okay, so I see your comment, Tiffany. It is not a reasonable position. So let me ask you this in the chat window. Why is it not reasonable to expect drugs to be 100% safe? Why is it not reasonable? Why are we establishing a standard for the drug companies that nobody on the planet Earth could ever reach? Okay, all right, I'm seeing some really good responses in the chat window. That's exactly right. If we give the exact same pill with the exact same medication at the exact same concentration to every person on the planet, some people are still going to get sick, right? And the reason is that while the pill is the same, the medication is the same, the personal DNA makeup of every human being is in fact different, which means different people with different genetic makeups will have different unique personal biochemistries, which cause them to metabolize the drugs differently. And in some ways, some people, unfortunately, it will have toxic side effects. It will make people sick, okay? So I'm trying to get to the issue of, why do we have ridiculously high drug costs? Yes, some of it is just plain old good old fashioned corruption, absolutely, without a doubt. But there's another side to that obvious answer. It's not just corruption, it's also that, as a society, I would make the argument that we pretty much want to have our cake and eat it too. I'm okay with the drug being on the market that hurts somebody else as long as it doesn't hurt me. Okay, I mean, that's the attitude I see out in the world. Okay, so I want you to think about how harmful that attitude might be. Okay. Each person metabolizes the same drug differently because we are all genetically different. Okay, last thing I'll make a comment on, then we're going to wrap up for today. Okay, question in the chat window. Here's the question. Here we go. How long on average do drug companies have legal patent protection on new drugs so that another company cannot copy it and sell it at a lower cost? Yep. Okay. There's some really good answers in there. And again, we're talking in generalities. In some circumstances, it's going to be a little less. Some circumstances, it's going to be a little more. But the typical average today for patent protection is about 17 years. 17 years of patent protection for a company to develop, bring a new drug to market, and be able to sell that free of competition so they can recover their investment of developing the drug, which we said is already in excess of $1 billion, so that they can make some profit. I mean, they are a company after all, and we live in a capitalistic world. Nobody's going to do this stuff for free, right? Nobody's going to do it for free. Okay. Okay. So if you take a look again at figure 20.6, at figure 20.6, you'll notice that panel A of figure 20.6 says natural penicillins, and panel B of figure 20.6 says semi-synthetic penicillins. And the point I want to make right now is that, you know, when a company is heavily into its 17-year of patent protection, that company is already thinking about, well, when will the patent protection expire? Which means another competing company could take the drug and manufacture it at a lower cost. To you and I, the consumer, we love that because we love our generic medications. Hey, I'm on a blood pressure medication. I get a 30-day supply for $5. I love it. It works for me. It helps me. And it's only $5. Obviously, if I had to pay $500 for that medication, I'd be a lot more upset, wouldn't I? But this is the kind of things we have to deal with at times. So when a patent is getting ready to expire, or in the years preceding that, what a drug company will do is it will take its current drug that has patent protection, and it will ask its scientists that are the employees. They'll say, hey, please go in and figure out a way that we can modify this molecule so it's a little bit different from the original molecule, but it's still effective at doing something like killing bacteria or whatever it might be. So they modify the molecule that they already have patent protection on, and then they reapply for a new patent with a slightly modified molecule. Now, usually with the slightly modified molecule, the US legal system might give them less patent protection on a slightly modified version of that drug, but they still might get another 10 years of patent protection on the newly modified drug, which is why, as you see in panel B of figure 20.6, you're going to get semi-synthetic penicillins, oxacillin, ampicillin, which are very chemically similar to penicillin G, but nonetheless a little bit different. Okay, a little bit different. So drug companies try to maintain their cash flows by modifying existing drugs and applying for new patent protection. Okay. All right, folks. So that's just a little bit of spiel on the legalities and the financial realities of drug development in the United States of America. There is tremendous corruption in our system, tremendous corruption. There's no reason drugs have to be the cost that they are in general. But I will say, from a technical perspective, it is very financially costly to develop and clinically test new drugs. And we do have to allow the drug companies to make some profit, or else nobody's going to do this work. Okay. Do you have any thoughts or questions you'd like to comment about this topic? Feel free to speak up. Okay, let me ask this question.
I have a comment.
Okay, Rachel.
When we were doing the lab and we were looking at new antibiotics, that were produced or put on the market in the last 3 years, I did find out that only 1 of every 5 get FDA approved.
Yeah, absolutely. Isn't that kind of scary? But it's absolutely the reality, right? It is more likely that a new drug that is in the process of being researched and developed, it is more likely that it will fail in clinical trials than it will succeed. So there's a very high failure rate of medications. As you said, only 1 in 5 get approved. So that means 4 out of 5 don't get approved, right? So the company's been spending $4 billion to develop 4 drugs that never get approved for sale in the United States. Wow, who's going to have to pay for that development cost? The consumer, because that cost is going to be pushed into the products that are sold, right? So that the company can keep rolling. It's a very ugly situation that we have, very ugly. Okay, so let me address a couple questions in the chat window. So Aubriana, your question is this: is it true that people cannot sue for bad reactions to COVID vaccines? I doubt that's true very much. I doubt that it's true that you cannot sue, but I would suspect that you would not be successful, because typically, when you get a vaccine, you also get a paper with the warnings on of what this drug or this vaccine might do to you, right? I mean, you're accepting a risk when you take the vaccine. I got the flu shot about a month ago, and I had a big paper with all the possible side effects and everything, right? So you're being forewarned about what this might do to you. And so if you choose to proceed, well, you chose to proceed. So you could sue, but I think the chances of you being very successful with that is pretty low. Yasmin, your question. So if everyone reacts to each medicine differently, how do they eventually approve it? Yasmin, the quick answer to that is that this is whether a drug should be approved or not is based on statistics and probability. Typically with a clinical trial that reaches stage 3 clinical trial, the company will actually test the drug on maybe a couple hundred people and watch those couple hundred people for bad reactions. And if, they will document certain side effects that have been observed and things like this. But if the side effects don't seem to be really horrible, really bad, and there does seem to be therapeutic value to the drug, in other words, it seems to do more good than it does more harm, then the drug still has a chance of actually getting FDA approval and being put on the market. If you think about it, if a drug passes through clinical trials and has done marvelous, really didn't make anybody sick and had a good therapeutic profile, that's magnificent. But that data to make the decision as to whether to sell it across the entire United States is being made on testing that drug in a few hundred people. Let's be crazy and say they do it on 1,000 people. Okay, so 1,000 people took the drug, they seemed to benefit from it, there was no horrible side effects. But how many people are in the United States of America, right? 350, 400 million people. Okay, so there's no way that amount of data can be truly predictive of how the drug will be metabolized in the body of 350 million other people. Okay, so it's a numbers game, it's a probability game, it's a statistics game. Okay.
Thank you.
Sure. Okay, Geneva, what if they mandate the vaccine for everyone? Would that change the outcome of winning a lawsuit? I suspect it would. Right? Politics is an ugly game, and it is what it is. Okay. Okay, so Tiffany, let me just read your comment. Also for the COVID shot, depending on what and when you got it, they had it in the disclaimer, whether people realized it or not, that it was not FDA approved and it is only authorized for a test administration. That being said, if you chose to partake in the clinical trial test of getting the shot before being FDA approved, some insurances won't cover it if something happened to you. Yeah, Absolutely. I don't have any arguments against that. You're absolutely right. So, you know, society is a mess. We're just never going to agree with each other, right? You get two people in a room, and they're going to disagree. That's just how life is. Okay. So anyway, okay. Thanks, Tiffany, for the comment. Any final questions before we switch gears, right? This is something we could talk about for a long time, isn't it? Because it's an interesting topic. Okay. Okay. Let's shift gears into lab.
I do have a quick question real quick. I'm sorry.
Okay.
So with the, like that you said how, you know, drugs have to go through a certain period before they can be administered and stuff like that. My question on that is, so they came up with all these different vaccines for COVID. So that means that this is something that they had been working on for years before that even COVID began, correct?
No. The actual vaccine development started once we realized, as a global civilization, once the World Health Organization was aware that there was a problem, then vaccine development began.
I know that Pfizer at one point was at first it was tweaked for to treat it in one specific thing, and then when COVID came around, they had adjusted it to help with COVID.
Yeah,
But
that's very
it was
okay.
being used for something else originally.
Okay. Yeah, I mean, absolutely. That very well might be possible. And in fact, I can tell you that there are coronavirus vaccines that have been on the market for many years for animals to treat dogs and things like that. The only reason I know that's because my wife is a veterinarian. So I don't discount what you're saying, Tiffany. I believe that, you know, a company might have had some previous research experience in this. And that might have helped them accelerate how they could respond to this global health crisis we had. Obviously, when the vaccine was put out, it was put out under emergency authorization, which means hey, we've got an emergency here. We need something to try to stem this situation. We haven't quite checked off all the check marks on our safety checks. We haven't crossed all the T's, dotted all the I's. But right now, it seems like an acceptable risk to put it out there to our communities. So it is one of those things, right? When you have an emergency, sometimes you cut corners. Okay. All right, folks. Thanks for your enthusiasm on this topic, right? This is something we could really go on for a long time. Okay, so I'm going to, let's see here. I'm going to stop recording because I'm stopping my official lecture here.